Welcome everyone, my name is Sebastian and today I would like to talk to you about our work on adversarial and graph inference and generation. Let's start with some background on de novo chemical design, where the task is to find molecules with certain properties. In this day and age, this could be finding an antiviral drug inhibiting SARS-CoV-2. De novo chemical design is very challenging because the space of molecules is huge and we can't compute the properties we are interested in for every existing molecule. Moreover, molecules are discrete in nature, which means we can't use gradient-based optimization to explore the space of molecules efficiently. In the last couple of years, people use deep generative models to address this problem by projecting molecules into a continuous latent space where gradient-based optimization can be performed. Here you can see an example of such a model, which takes in a molecule, represented as a graph, and projects it into a latent space. The decoder then takes the latent representation and outputs a graph representation of a molecule again. During training, we want to minimize the KL divergence between the latent space and the prior distribution, and we want to minimize the distance between the input and the output graph. Now, because input and outputs are graphs, this means we need to solve an expensive graph isomorphism problem in order to compute this reconstruction loss. If you look at prior work in this area, then the encoder is usually a type of graph convolutional neural network. For the decoder, there are two options. We can either use a set of fully connected layers to generate a graph in a single step, or we can use a recurrent neural network to generate a graph sequentially, one atom at a time. When focusing on the type of generative model, then the majority of previous work is likelihood-based and requires computing a reconstruction loss. They try to mitigate the graph isomorphism problem by traversing nodes in a fixed order, sampling a couple of adjacent symmetries corresponding to the same molecule, or performing graph matching. There's one approach called Mulligan, which uses an adversarial loss, but it has no encoder and can only generate molecules. In other areas of machine learning, people proposed adversarial learned inference and extensionalis, which do not require optimizing an explicit reconstruction loss to train an encoder-decoder pair. However, these approaches are only applicable to continuous valid data, such as images, and not to discrete data, such as graphs we are dealing with here. In this work, we propose adversarial learned molecular graph inference and generation, or ALMGIG, which does not require solving an expensive graph isomorphism problem. Our encoder is an extension of the recently proposed graph isomorphism network to multigraphs, and our generator uses the global softmax trick to generate discrete graphs. To generate chemically valid graphs, we enforce connectivity constraints during training. Finally, we show that current evaluation metrics are flawed and propose a set of improved evaluation metrics to assess the distribution learning capabilities of methods. To understand our optimization scheme, I'll start by briefly explaining adversarial learned inference. The setup is similar to a graph variational autoencoder, except that what is called decoder is now called generator, and instead of a reconstruction loss, an adversarial game is played. In this game, encoder and generator try to fool the discriminator such that it cannot distinguish between two joint distributions anymore. The first joint distribution is the joint distribution which has two parts. It's the encoder joint distribution. The first part is over the real data, which is marked in green, and the second part is the distribution over the latent space produced by the encoder, marked in blue. The second distribution is the decoder joint distribution, with again two parts. The first part is the prior distribution over the latent space, marked in green, and the second part is the distribution over graphs produced by the generator, marked in blue. The training objective is to match these two joint distributions. However, we can see from the figure that the reconstructed molecule via the red path is not constrained. This problem has been addressed by Alice, which adds a second discriminator that distinguishes between a pair of real graphs and the real graph and the reconstruction. Now, encoder and generator have to fool both discriminators. They show in the papers that at the equilibrium of this adversarial game, both joint distributions match, and the input matches the reconstruction. Unfortunately, if encoder and decoder and discriminators are deep neural networks, getting close to the equilibrium is rather hard. We address this problem by providing an additional signal during training, namely that of a unary discriminator as in a standard GAN. You will see later on that this allows us to capture the distribution over the molecules more accurately. The resulting adversarial game involves 3 discriminators that encoder and generator try to fool. The first one is the unary discriminator, distinguishing real from randomly generated graphs. The second discriminator is the joint discriminator, distinguishing 2 joint distributions over graphs and latent variables. And finally, the cycle discriminator, which distinguishes a real graph from its reconstruction and is ultimately the key to avoiding the graph isomorphism problem. Moving on to experiments. We used the QM9 dataset containing molecules with at most 9 atoms. We compared against 2 state-of-the-art graph variational autoencoders, which use an RNN-based decoder and enforce valence constraints such that generated graphs are always chemically valid. We also compared against Grammar VA that works on string representation of molecules. The only existing adversarial approach is Molgan, which we included as well. Finally, we added a random baseline, which chooses atoms and bonds randomly, but always in a way such that the graph is chemically valid. Before diving into the results, it is important to have a closer look at the evaluation metrics that have been previously used. Those are validity, uniqueness, and novelty. If I want to have a perfect validity, all I need to know is how to produce one single molecule and just do that over and over again, as it's depicted here. If I want to have a perfect uniqueness, I can just start with an arbitrary molecule and extend it by appending one carbon after another. If I also want perfect novelty, I would search the training data for the heaviest or biggest molecule and just extend that one by adding one carbon after another. This shows that a very simple approach can already lead to perfect validity, uniqueness, and novelty. And clearly, if these metrics can be footed easily, they surely cannot capture whether a model actually learned something useful. So let's take a step back and think about what question we actually want to ask during evaluation. We want to know whether a model can generate chemically meaningful molecules with similar properties as the molecules in the training data. This has been acknowledged by Brown et al., who proposed to compare the distribution of 10 chemical descriptors in terms of KL divergence. Unfortunately, the KL divergence has some issues, and we propose to use the Earth-Smoothest distance, also called von Wasserstein distance, instead. First of all, the Earth-Smoothest distance is a metric and satisfies symmetry and triangle inequality, which the KL divergence does not. But more importantly, only the Earth-Smoothest distance does quantify spatial shift. So if you look at the example on the left, the KL divergence would only consider the part where P and Q overlap, but how far the remaining part of Q is from P is irrelevant. Even worse, when P and Q do not overlap, as shown on the left, the KL divergence is undefined. Now that we have a nice evaluation metric to assess how well-generated molecules represent the properties of the training data, let's have a look at the results. You can see that the proposed AMG captures the distribution of the molecules most accurately, closely followed by CGVA. After that, there's a considerable drop in performance. The nice part is that we can now look at individual properties to understand why AMG is better than CGVA. The first row shows molecular weight and clearly reveals that CGVA creates molecules that are heavier than what is in the training data. This is not the case for AMG. Moreover, CGVA creates molecules with bigger aliphatic rings, as shown in the second row. Finally, if we zoom in on this part here, we can see that there are 2 small modes containing about 2% of molecules. These lighter molecules are better captured by CGVA. Moving on to the different adversarial learning schemes. Here, we compare the same architecture trained by playing different adversarial games. The results show that AMG with its 3 discriminators is clearly superior to the other schemes. If you want to understand why, we can have a look at individual distributions again. When looking at molecular weight for Alice, which has no unary discriminator, we can see that it suffers from mode collapse. Alice only captures heavy molecules with almost no aromatic rings. To summarize, we proposed AMG, which avoids solving an expensive graph isomorphism problem during training by enforcing the reconstruction property implicitly via cycle consistency. This results in AMG more accurately representing the distribution over the space of molecules than previous methods. We also showed that common validation metrics are unsuitable. Instead, we proposed a new set of metrics which can reveal valuable insight into what type of molecules a model can and cannot generate. If you want to use AlmGeek in your research, you can check out our GitHub repository, which contains all the code to reproduce our results. Thank you for your attention.